Hello everyone, I'm here to present our work called Multimodal Sheaf-based Network for Glioblastoma Molecular Subtype Prediction, which was done within the Basira lab. Glioblastoma is the most aggressive form of brain tumor with a median survival of just 12 to 16 months. The accurate diagnosis involves combining multiparametric MRI with invasive biopsy for histology and, when available, molecular sequencing. MRI offers microscopic information like tumor structure and location, while histology captures some genetical insights and the molecular subtype. While MRI and histology provide complementary and vital information for treatment planning, it's often that histology and molecular sequencing are not always accessible, which can impact clinical decisions. The multimodal integration in the biomedical domain remains limited. When combining MRI and histology, the three main challenges arise. First, spatial and scale mismatch, the heterogeneous nature of the data, and the incomplete or missing modalities. These challenges hinder conventional deep learning methods, as they all assume the features are uniformly aligned, while in practice, co-registration between MRI and histology is often missing. To address these challenges, we use graphs that can represent the data as nodes and edges, making it possible to model complex spatially distributed structures, meaning we can capture relationships between our modalities. However, standard GNNs come with their own limitations. A major issue is the oversmoothing. As message passing layers stack, node features become increasingly similar, losing their distinct characteristics, which is particularly problematic for our case. To overcome the oversmoothing problem, we anchor our model in sheaf theory, a mathematical framework for systematically combining local data into a consistent global picture. Imagine a topological space X with open sets U and V and the overlap between them. In our case, U and V represent MRI and histology, two local views of the same tumor, each capturing different aspects. A sheaf F assigns data, here continuous functions, to each open set. Its key property is the local consistency, which means that in the overlapping region, the data from both sets must agree. Enforcing this consistency across overlaps allows us to reconstruct a coherent global structure. If we view a graph as a discrete manifold, the nodes act as local spaces and the edges as their overlaps. Restriction maps are linear transformations from nodes to edges which project features into the shared space, ensuring local consistency and alignment. Using sheaf neural networks lets us naturally model how heterogeneous node features contribute to a shared overlapping space while preserving their distinct characteristics. The existing methods include co-attention fusion, often requiring paired and aligned data, the feature concatenation after GCN processing, which typically lacks deep interaction between modality features, and contrasting learning, although flexible, still rely on alignment assumptions in the shared space. To overcome these constraints, we propose a multimodal sheaf-based network. Our proposed model multimodal sheaf-based network, or MMSN for short, consists of three main components like MRI and histology encoders, as well as the sheaf neural network and the prediction stage. Before diving into the algorithm itself, let's walk through how we transform the raw images into graphs. For MRI, we begin with the 3D scans for each subject, and using the annotated tumor mask, we identify the tumor region. Within this region, we randomly sample voxels and extract radiomic features. By treating each voxel as a node, we form a graph where edges are based on k nearest neighbors. And as for histology, we process the whole slide images by extracting smaller tiles in accordance with the annotated masks. The tiles become nodes in the histology graph with edges defined in a similar spatial manner. Each tile is then passed through a CNN, in our case a ResNet, to obtain high-level feature embeddings. Starting with the input graphs constructed from MRI voxels and histology tiles, we use the regional labels to aggregate node features within each region. These aggregated features are then passed through a GCN, updating the features and effectively producing a region-level graphs. Next, to fuse the modality features into a single graph without prior alignment, we start from the region-level graphs for each modality and project their features onto a shared latent space. This latent space is represented by a graph with its own learnable features. We apply a softmax function over the features from both of the modalities and the latent graph to generate the assignment ways. that control how much each modality's features contribute to each node in the latent graph. Following the aggregation, the shared updated graph is formed that captures tumors' underlying topology. Next, we apply the Sheaf neural network on the updated latent graph. Using learnable restriction maps, we introduce a Sheaf-based message passing. We produce a graph embedding through a linear layer with activation, which is then used to predict molecular subtype. To train the model, the loss combines two parts, the classification loss and the consistency loss, enforcing the nodes connected by an edge maintain similar views in the shared edge space. We also evaluated our model's ability to handle missing modalities. We can use the available modality to define the edge space and then reconstruct the missing modality's node features by projecting back from this defined edge space. And to support this during training, we introduced a reconstruction loss that minimizes the difference between the original missing node features and their reconstructions. Datasets with paired multimodal glioblastoma data are extremely scarce. For this study, we used the only available open-source dataset, the IVGAP study, which includes data from 41 patients. We also benchmarked our model against several state-of-the-art multimodal approaches, and all models were trained and evaluated using threefold cross-validation and early stopping to prevent overfitting. Our results demonstrate consistent outperformance in comparison to the benchmarks. We observe a greater increase in sensitivity than specificity, which we explain by the heterogeneous nature of the glioblastoma, meaning that overpredicting the presence of a subtype is more tolerable than missing it entirely. The TSNE visualization of class separation shows less dense clustering comparing to MHAN, which we interpret as the model's ability to capture mixed or overlapping subtypes within individual patients. Our model also shows consistent though variable outperformance compared to the adversarial attention-based variational graph autoencoder in the scenario of missing histology modality. To conclude, our model effectively fuses two diagnostically significant modalities, and by employing the Sheaf neural networks, our model overcomes the oversmoothing problem and effectively integrates complementary information from both modalities. This work contributes to advancing the development of virtual biopsy techniques, which could reduce reliance on invasive procedures. Future work will focus on evaluating the model on larger datasets and exploring the integration of other modalities. Thank you very much.